Okay, so again, when the stapes plunges in to the oval window, it causes the perilymph to move in the upper chamber. That perilymph then causes the endolymph in the cochlear duct to move. That causes the perilymph in the lower chamber to move, okay, in this direction. And when that endolymph moves in the cochlear duct, it causes the hair cells to get pushed up against that tectoral membrane, and it causes deflection of those hair cells. And deflection of those hair cells can lead to the depolarization, the release of neurotransmitter, and then the uh, and then the neurons that are associated with each hair cell will then send action potentials through the cochlear branch of the vestibular cochlear nerve. Now, we talked about some structures of the brain. So the vestibular cochlear nerve is actually attached to the brain on the medulla oblongata. So this auditory information would have to then travel through the medulla oblongata, and we ultimately want to get it to the auditory cortex where? In the temporal lobes. So it would have to travel through medulla oblongata, what's directly above that, the pons, then through what's directly above the pons, it would have to go through the midbrain. Then it would have to go to the thalamus before getting to the auditory cortex of the temporal lobe, because the thalamus is a relay center which takes all of the sensory information and decides where it needs to go, right? Which particular lobe it needs to send that information to. So then it would go to the thalamus. The thalamus would decide that it's auditory information and would send it to the primary auditory cortex, because the primary auditory cortex just is responsible for bringing auditory information in. Then it would have to go to the auditory association to be analyzed for it to decide what it means and your judgment and emotions towards what you're hearing. Okay? So now let's move on to the organs of equilibrium and balance. So here, this is the vestibule, that chunky region. This would be the cochlea, right, where you would find the organ of Corti, those hair cells that would send information through the cochlear branch of the vestibular cochlear nerve. This is the vestibule, and when you think vestibule, you want to think static equilibrium. Okay, and static equilibrium, it senses the position of the head. The organs associated with the static equilibrium sense the position of the head, maintaining a stability and posture when the head and body are still. When you think semicircular canals, you are going to think dynamic equilibrium. And dynamic equilibrium detects motion and aids in maintaining balance while in motion. Okay? And the information from the vestibule and the semicircular canals would be sent to the brain via the vestibular branch of the vestibular cochlear nerve. And so here, when you look in the vestibule, there are two enlarged regions. There's this region called the utricle and this region called the saccule. And contained in the utricle and saccule are the organs of static equilibrium, which are called the macula. So maculae for plural. Okay, so this is a macula. And the structure is, here we have our hair cells, and those hair cells have the stereocilia that stick up into this gelatinous matrix. Okay, so the stereocilia stick up into the gelatinous matrix, and how action potentials are going to get sent along these nerves will still be the deflection of these stereocilia, but because they're in the macula, it's sending information with balance and equilibrium. The hair cells in the organ of Corti of the cochlea send auditory information. And so if you look at the gelatinous matrix above the stereocilia of these hair cells, you see these rock-like structures. These are calcium carbonate crystals called otoliths. And the otoliths just give it weight, give the structure weight so that you can have a greater response to gravity, to movement. Okay, you can see that when the person tilts her head forward, these otoliths, these calcium carbonate crystals, fall forward, tugging on that gelatinous matrix, deflecting the stereocilia of the hair cells, and then causing action potentials to be sent along these neurons to the brain, dealing with the position of the head. Okay, and so these are the organs of static equilibrium called the maculae. The hair cells with the stereocilia embedded in a gelatinous matrix with otoliths above that. And so when the head changes position, those otoliths will fall in a particular direction, tugging on the gelatinous matrix, tugging on those stereocilia, and deflecting them one way or the other, causing action potentials to be sent along the vestibular branch of the vestibular cochlear nerve. Now back to, so here is the vestibule again. And attached to the vestibule are these half-circle structures called the semicircular canals. And where the semicircular canals attach to the vestibule are these enlarged regions called ampulla. And contained within these ampulla are the organs of dynamic equilibrium. And so, cool thing about these semicircular canals are that they're located at right angles to each other, and so that allows us to sense movement in all three planes. So think 3 dimensions, 3D, 3 planes. These are all right angles to one another, so for every plane. Sense direction, movement in 3 directions, 3 planes. And so here, this is the enlarged region called the ampulla, and the fluid that's going to be moving through the semicircular canals is the endolymph, which we talked about is present in the cochlear duct as well. And so here, this structure of dynamic equilibrium, this is called a crista ampullaris. So the whole thing is called a crista ampullaris. And the structure is, we have our hair cells, and the stereocilia are projected up into this gelatinous structure called the cupula. Okay, the cupula. And when you're moving, what happens is this endolymph will move in the direction opposite of what you are moving, right? And so this endolymph flows, pushing this cupula one way or the other, depending on which way you're moving. And when it moves this cupula, it deflects the stereocilia of the hair cells, causing action potentials to be sent along the nerves, the neurons that are associated with the hair cells. This information would also be sent out via the vestibular branch of the vestibular cochlear nerve. Okay? So when the endolymph moves over the cupula, this gelatinous matrix, it deflects the stereocilia of the hair cells that are projected up into this gelatinous matrix, causing action potentials to be sent. Now, the vestibular branch of the vestibular cochlear nerve the vestibular cochlear nerve is attached to the brain at the medulla oblongata. So this information would go to the medulla oblongata, then to the pons, and the pons would relay that information to the cerebellum, because this is balance and equilibrium. It goes to the cerebellum. Information from the eyes as well would be sent to the cerebellum, because our eyes help with determination of equilibrium and balance as well. And all of that information would be integrated in the cerebellum, and then the cerebellum would send out information back to the appropriate skeletal muscles to contract or relax to help maintain equilibrium, balance, and posture. So this information from the vestibular branch would ultimately go to the cerebellum.